In this video, we're going to talk about the biomolecules, continuing on with our chemistry. And the biomolecules are just basically the four categories of molecules that make up all living things. So let's dive in. These are the key terms. And we're going to start off with a reminder. We'll hit this a little bit harder in class when we meet, but remember that in biology, one of our themes is that structure informs us of the function. So every shape evolved for a specific purpose, even down to the chemicals. We can infer what a chemical's purpose and role in a cell is going to be based on its overall chemical structure. Whether it's polar, whether it's nonpolar, what its overall shape is, its size, all of these things determine how the molecules interact and behave with each other, which puts them into a specific role within the cell. So these biomolecules vary in size. However, we often also call them macromolecules. So biomolecules means life molecule, macro means large molecule. And that's because the way these molecules are made basically puts no upper limit to their size. They're made up of repeated basic chemical structures called monomers. These are these guys right here. And what we do is we take this base structure and we just attach it to itself over and over and over again to create what's called a polymer. This whole process of sticking them together is polymerization, or creating the polymer. So mono means one, mer means unit, and poly means many. So the monomer is the one unit, and the polymer is the multiple unit. So the way we make these macromolecules or large molecules is just by continually adding on more and more monomers as needed. Now, these monomers are always connected together with covalent bonds. So these molecules tend to be quite strong in their associations, and it takes a lot to break one monomer away from the group. But this is the basic template that you're going to see over and over for these four groups of molecules that we're about to talk about. Now, water is the key to these polymerization reactions. If we didn't think water was cool enough after what we just learned, it's even cooler because the way that we add or separate the monomers from each other is entirely a water-based reaction. So to bring two monomers together and attach them, we do a dehydration synthesis reaction. So this reaction combines the monomers, and in doing so, the bond that is created kicks off a hydroxyl, or OH, and an hydrogen, or H. And when those two get broken off from the whole set of molecules here, they form together to make a water. Now, the opposite is true if we want to break apart two monomers that are part of a polymer. If we want to break a polymer into pieces, we take that water molecule and we force it back into that area that they once belonged to. And when that happens, we can then stabilize each monomer by adding the hydroxyl and the hydrogen to it. So if we look at the terms, dehydration, we're removing a water, we're taking water away. Synthesis means to make something. So we are making by dehydrating. And hydrolysis, hydro means water, lysis means to break. So a hydrolysis reaction is using water to break the bond. So we often will do dehydration synthesis if we want to copy our DNA or make muscle. But if we want to digest our food, we'll use hydrolysis, things like that. So the four biomolecules or macromolecules that we're going to talk about are lipids, nucleic acids, carbohydrates, and proteins. And most of these should be familiar to you just in general, because at least proteins, carbs, and lipids, or fats, those are on our nutrient labels, because basically everything we eat comes from a living thing, either a plant or an animal. And if these are the four molecules that make up all life, we should expect to see them. Now, we don't get any nutritional value from nucleic acids, and our bodies don't really break them down. So that's why we don't include them on our nutrition facts label. But let's briefly dive into these four. So we're going to start with carbs because I think they are the most familiar for us to talk about. And carbohydrates, or carbs for short, are the macromolecule that the body uses to provide energy for the cell. So we can take these simple sugars and break them down and use them for energy. We'll specifically talk about glucose because glucose goes into our mitochondria and we use it to create ATP, or energy molecules, for the cell. But carbohydrates can also make these really large, very complex structures that have strong lattices and things like that. And they can be used to store energy long term in plants, or to even create structure in plants and animals. So something like cellulose or fiber is in plants, and it helps make up the structure of their leaves and their stems and their bark and all of that. Glycogen in our muscles is used to kind of as an intermediate between a fat storage and using the energy right away. So if we want to store the glucose but be able to access it much faster than we can from fat, we will store it as glycogen. Starch is effectively fat for plants, so that's how they store their energy long term. You have chitin, which is basically the carbohydrate that's found inside the shells and the carapace of our insects and our crustaceans, everything that's an invertebrate, where they need protection outside because they don't have a skeleton inside. And then bacteria, their cell wall is made out of peptidoglycan, which is a sugar and a protein mixed together. Now, carbohydrates have a couple different names that we ascribe to them based on their size. The monomer, remember the building block, because all of these are biomolecules, macromolecules. We start with a monomer and then we just attach it over and over again to make a larger version of it. So the monomer is called a monosaccharide. This could be glucose is an example. Galactose is another example. So the monomer is the monosaccharide. Mono meaning one, saccharide meaning sugar. So this is the one sugar, the base form. Then our simple sugars are those that are like monosaccharides or disaccharides. Sometimes we can attach two of the monomers together to form a disaccharide, di meaning two. In this case, you might have an example of something like sucrose or lactose. And then there are the complex sugars. These are the polysaccharides. You may have also heard the term oligosaccharides. That's kind of a long carbohydrate, but not quite as long as a polysaccharide. So poly just means many sugar. And so this can be hundreds and hundreds of monomers connected together. An example might be starch. And then we have nucleic acids. We're only going to briefly touch on them here because when we talk about DNA, we're going to get much more in depth into their form then, so we can directly apply it to how DNA works. So DNA, or nucleic acids in general, they are the cell's informational molecules. They are also the cell's energy storage molecules. So the monomer of a nucleic acid, the building block, is a nucleotide. It'll have a phosphate group, a sugar, and some kind of nitrogen base. Now, in DNA and RNA, where we're trying to store information in a code, we connect those together to make long chains, and then we can attach two chains together. But sometimes we leave it in this base form and add a couple more phosphates to it, and then we call it ATP, and that is the cell's energy molecule. So those are all examples of nucleic acids. And just remember that the nucleic acid, the monomer, is the nucleotide for it. Then we have lipids. These are the cell's energy storage and protection molecules. So ATP doesn't exactly store energy, it's the functional energy of a cell. Really, this is like we're storing it in the battery and we're going to use it later, but the battery is not the active energy. I know that that's not how it really works in battery usage, but think that we have to convert it maybe from the oil form. Oil and fossil fuels have energy. This is kind of like the lipid. And then we put it in the battery, which is like the glycogen or the ATP, and we use it directly. That's a better example. So, lipids are large, nonpolar molecules. They're often primarily made of hydrogen and carbon. They tend to be carbon backbones, absolutely surrounded by hydrogens, and we use them to store our energy long term. We can also use them for protection. The reason why you don't want to have 0% body fat is because we actually need fat to protect ourselves. It helps prevent friction against your bones and muscles to the rest of your body. It's quite painful to be born with the disease where you cannot have any body fat. So the molecules that fall under the lipid category are fats, oils, waxes, and steroids. And so these guys technically have no true monomer or polymer form. And that's because they can take a lot of different forms. We can have triglycerides, we can have phospholipids that make up our cell membrane, we can have this basic steroid structure that becomes all of our hormones and things like vitamin D and cortisol. So there's a lot of different structures that lipids can take. So, of all four of them, lipids are the ones that have no true monomer, but they tend to be hydrocarbons, where the carbon and hydrogens are the backbone. And saving the best for last is proteins. We are going to very, very briefly touch on them now, and we're going to dive a little bit deeper into them next class. So, proteins are the most numerous and the most versatile biomolecule in the cell. They have so many different forms that they can take, and they're really important to chemical reactions, to the structure of our cells, to cell signaling. They help make up certain kinds of hormones. So proteins are incredibly important to the cell. And they're basically just polymers that are made up of amino acids joined together by peptide bonds. I'll get into what those terms mean more in a second. They have the most unique structures, and because structure tells us what the function is, that means they're going to have the most unique functions in the cell as well. These functions are numerous. They make up our enzymes. They help us form little channels in our membrane to help get materials in and out of the cell. They form hormones like insulin. They create toxins in a lot of different organisms. They form our muscle fibers. They do so many different things. They help us create our copies of DNA. We're going to talk a lot about proteins throughout this semester. And so proteins, their monomer is the amino acid. So amino is based on a chemical substructure that is this amine group right here, and we'll get a little bit more into that in class. But the overall structure of an amino acid is this right here. So we have one amino acid here, another here, and what happens is they get connected together with a peptide bond. So through that dehydration synthesis reaction, we form a special type of covalent bond that we call a peptide bond. And a peptide bond is just the name that we give to the covalent bond between amino acids. Think P for peptide, P for protein. That's the way I keep it straight in my head. And you may have heard of a ribosome. This is one of our special organelles inside the cell. And it is the thing that helps create that peptide bond. And this is going to be important when we talk about it later on in the semester. And we're going to leave it there and dive a little bit more into all of these in class together. Don't forget to study for your quiz on ecology, and don't forget to submit your topic choice for the bioengineering project as soon as you can. See you guys soon.